Okay. Um, Thank you everyone for joining once again. This is going to be our last session with prepared material where we will actually talk about some of the host pathogen interaction project ideas. We'll talk about kind of a summary of what we have learned throughout the program. And we'll talk a little bit more in detail about protein-protein interaction and host response and different immune response factors. Also, how this can be applied to clinical situations for drug discovery, for vaccine design, and other topics related to patients. So before we do that, I do want to mention that there has been quite a lot of activity on the platform throughout this whole program, and I think it's very exciting for us to see how much you have been able to accomplish in terms of completing different courses and being able to translate some of the course material and the sessions that we've had together into project proposals. So we do have a number of different project proposals that we'll review towards the end. I think that we also leveraged a lot of the different elements of the program very effectively by having a lot of interaction throughout the different issues that are coming up. As you know, technology can be very good and sometimes also cause issues that we have to work through. So I thought that it was an overall good experience being able to identify challenges that came up and address them. And hopefully most of you have been able to resolve some of the issues that have come up. Okay, yes, and we will talk to the topic of project proposals. You do have some time still, so let's review the program overall, and we'll get to the projects towards the end of the program, but yes, you still have time. Okay, so on the forum, I just want to make sure that all of you have been to the forum to review some of the session recordings and some of the answers to the questions that you've posed. We also have some guidelines for project submission. We'll be adding more information to there over the weekend. So make sure that you review that. And to kind of see where we are as a group, let's also review the program overall so that we remember what kinds of topics were covered in different sessions and how they apply to the problem of finding a project data set and developing a project based on some of the assignments and some of the material that we've gone throughout this program. So as you remember, we started from talking about next generation sequencing, how to understand the different file formats, align reads to the reference genome, annotate those elements that we find, and find different aspects of pipeline output, including the output that is intended for analysis or output that could be analyzed in different ways, like non-map reads, that we can then take and align to a database of viral genomes. So I hope that from that session, what you've learned is that we have many different types of next generation sequencing techniques. This is an important part to remember as you start looking at the different projects, because there's a lot of FASTA and FASTQ and BAM and SAM files and all kinds of variations in terms of how that data was produced. So paying attention to exactly which instrument and what type of information is in those files is going to be important for you to start applying the different analysis techniques. I think that it's also important that there's a huge amount of data out there, and this data represents a big opportunity for us to discover novel things that have not been reported, that have not been published. And I think that first project that we asked all of you to try and go through illustrates that kind of a problem. Many different cell lines are being used for development of biologics, which could include vaccines. And sometimes those can get contaminated with viruses. So it's important to review how some of this material that is available online can point us in that direction. After that, we talked about multiple sequence alignment. When we identify a genomic sequence and we can compare that sequence to a different sequence or even multiple sequences, looking for variation and looking for consensus sequences. Also understanding the quality of consensus sequencing. When we find a consensus genome, what does that really represent in terms of relationships on one hand, and also in terms of what kinds of an agreement do we see and how much of that agreement is reliable or non-reliable. Then we talked about, we did some practice analysis where we looked at different types of comparisons between FASTA files and FASTQ files, where we talked about how we can align multiple sequences. We can also compare different consensus sequences, or we can look for variation that is contained within FASTQ files. And we looked at some of the setting up some of the pipelines, finding the data, making sure that the data is organized in a proper way. And we know where to find metadata about each data set, so we know where it came from, and find things like geolocation, or even consider different sizes and different coverage that is found for different files. We then talked about comparison of different workflows, so essentially how we can leverage some of the different sections that we have on the platform, but also how do those translate to publicly available types of methods. Sorry, somebody's okay. So if you could please keep your microphones muted. Thank you. Okay, so we talked about different pipelines and looked at the different outputs and different results. And hopefully you've, throughout this whole program, you've been able to gain experience in analyzing different types of data sets, finding the right outputs, and extracting the most information out of those outputs so that you can get closer to biological interpretation. After that, we started talking about how an infection caused by a virus can develop into these pandemic or outbreaks. How there are many factors that play into that, and we can study those different factors looking at data collected from host species that could be reservoirs for these pandemic outbreaks, how we can look at also different patient populations and then study the virus development over time, how it acquires the capability to spread rapidly between humans, and also how that causes disease symptoms that are severe. So we then talked about disease progression and how the different types of phenotypes associated with disease develop as a result of different types of viruses. We started talking about different viral proteins that acquire specific mutations, and those mutations could lead to novel types of phenotypes and disease, what we call disease, right? Then we talked about how we can identify some of the factors related to the origins of human infection with coronaviruses, and looked at the comparison between hosts, intermediate hosts, and humans. And then also we could apply the same idea to multiple types of comparisons. And so I hope that from that we saw a way to find data and present analysis results and conclude from those analysis results in specific ways how potential routes of transmission and origin could be inferred. Okay, question, I got problem to visualize the output of my result. Can I go through specific software to resolve this problem? Yes, definitely. So if you could, again, post that question on the forum, and I'd be happy to, post an answer there. I think for this session, we have a lot of things to go through, so I'll try to if we have time, but if not, please post it on the forum. Okay, so then we started talking about different types of mutation variants, how we can look at different properties of mutations and think about how those properties affect the 3D structure of proteins and how we can map those and understand what impact that might have, for example, on binding to cell receptors, or also in general, how we can start understanding by looking at mutation variants, what types of mutation variants could be beneficial or detrimental for the viral adaptation. That type of information has different types of significance, and that different types of significance could be for vaccine design. When we think of areas that have a lot of variation, does that area on the protein, for example, have an impact for a vaccine? A good example for that is the Ebola virus vaccine that was designed, and it was designed around a specific change in the glycoprotein of the Ebola virus that seemed to have increased infectivity. So knowing that specific variation like that is going to have an impact on the pathogenesis of disease and also on prevalence of that specific disease helps understand how that disease could be, what kinds of interventions could be designed. And finally, today, what we'll talk about is host pathogen interaction. So we'll talk about how the host interacting with, how the pathogen interacting with the host really interacts at the level of proteins. We'll talk a little bit more about how complex those proteins encoded in the viral genome can be by making complexes of proteins, and how those actually cause and modulate the host response, like the immune response, as well as how those can cause specific types of reaction that are going to be specific to different people, different patients. And we'll do that by looking at specific types of data. So up until now, we dealt a lot with genomic data. We looked at how to find mutations, how to align genomic sequences. And we also asked you a few sessions ago to start looking at RNA-seq, how to analyze gene expression and isoform expression data. And so this session is going to deal with some of those types of data sets. So I wanted to provide you with a quick overview of RNA-seq. So this information on RNA-seq is all included in Transcriptomics 1, 2, and 3. So we'll briefly talk about that material to kind of connect the dots, because I also saw that a lot of the questions and a lot of the different pipelines that people have been trying to run maybe ran into some difficulties or conceptual disconnects between the methods and why those methods are useful and what types of problems do they really solve. So to give you an overview of RNA-seq, basically RNA-seq is separated into two parts. There's the processing part, and then there's the downstream analysis part. So let's start with the processing part. What's important when we do processing of reads is to remember that we take sequences of short reads. Those sequences of short reads a lot of times have been prepared using very specific techniques. And so it's important to clean up those reads. We don't take all of the reads because they can contain a lot of junk, and they can also contain a lot of variation that is technical in nature. It is not representative of the biological condition that we are trying to study. So pre-processing is an important step, especially for highly expressed genes that we will study later that become significant. And so that's why removing those technical variations is going to be important. The second part is mapping. There are a number of different mapping techniques, and the main idea here is that we have genes that consist of exons, and a big part of the translation, the transcription, process is when a specific gene sequence is alternatively spliced. And that alternative splicing appears to have a major role in a number of different conditions. So when we map on known transcripts, we are essentially forgetting about the possibility of that alternative splicing variation to be present in our data. The alternatives to mapping on known sets of transcripts, which might be a lot faster if that part of information is not relevant, but the alternative is to really map all of the reads on the reference genome to identify novel transcripts and all the variations of transcripts, essentially preparing a transcriptome. And that transcriptome could be then used as an alignment reference instead of the genome. Finally, after that step, we need to quantify the level of expression. Estimation of level of expression is not a straightforward task because those reads are not going to have a very consistent coverage. And so to say that a transcript or a gene is expressed at a certain number is not going to be so trivial. So the way that it is done is using different types of fairly complex algorithms that utilize a model that has relevance for how different sections are covered already and how to detect real variation that is statistically significant. Also, it's important to utilize different types of normalization techniques, but it's important also to remember which ones were used, because sometimes when we do downstream analysis, this type of normalization will interfere with the types of analyses that we would like to do. For example, differential expression is going to require raw sequencing counts, so read counts, instead of some normalization like FPKM or TPM that includes normalization for the total number of reads and for the length of genes in some cases. Okay, a quick question. Does the pre-processing applies also for data that we find on databases like NCBI, or only when we do sequencing for experiments? No, well, NCBI contains data. If we're talking about FASTQ files that contain individual reads, that was just deposited by whoever produced the experiment. So whether you made the experiment or somebody else made the experiment, they still use a specific library. They still use PCR breach amplification. And so those things are going to be important to remove and only take reads of high quality. And so those trim-o-matic and PCR removal of duplicates are going to be important in either case, because oftentimes we don't know exactly how those reads were generated. Okay, so a quick overview of a processing pipeline. We take those pre-processing steps. We utilize some kind of mapping techniques that might include mapping on transcripts or a combined strategy of mapping on the genome and then identification of novel isoforms, and then some form of quantification. There's a number of different methods to do that. Finally, what do we get at the end of this pipeline? At the end of this pipeline, we get a table of expression that looks something like this. For example, here in this output, you can see genes for the human genome. ENSG is an ensemble ID of the human genome. You see that it is actually different transcripts of that gene. 0.13 means this is a transcript of this gene. And then here you can also see in the column names that this is already cleaned using trim-o-matic. For example, no adapters, no PCR. And you can see that this was produced using paired end reads. Here you see some numbers, and these numbers are going to be important for us to understand, just even oftentimes by looking at them. Okay, for example, we have different distribution of data, right? And a lot of you have looked at quantile normalization and transformation of this table to log normal scale. So those types of transformations are going to change the numbers that you see inside. Oftentimes, you will see that these are not full numbers, so 2.9 or 51.78, and that right away tells you that there was some level of normalization here already. Okay, difference between cuff links and cuff merge. Well, if you look right here, this tells you exactly what these do. So cuff links is isoform construction. So you actually construct how an isoform or a gene transcript consists of what different exons. And cuff merge adds this information to the GTF file. The GTF file is then used to map those reads on transcripts. Okay, so we have this gene expression table, and after that gene expression table is obtained, we can add a downstream, a number of downstream types of analyses. For example, differential gene expression. But differential gene expression can also work just if you have a table of expression. So when you produce the table of expression yourself, it's important to remember, again, that you use raw read counts instead of TPM and FPKM. If you find that table of expression somewhere else, also look for what exactly are the units that I see here. And so then, once you've got your right types of table of expression, so you have your samples and expression of genes, those genes have to be the same across all of the samples that you are going to compare. And then you can organize them by groups. For example, in each group, you can have a minimum of 3 samples, and then you can compare conditions that those samples represent. Obviously, doing exploratory analysis for that is going to be important as well. But here you can find, for example, a workflow where you can just add those steps at the end of the same pipeline and add things like DE-seq, so differential expression of genes. How do we find those? RSEM, or EDGE-R or CuffDiff or some of these other techniques. And all of their outputs are actually going to allow us to differentiate between statistically significant and statistically insignificant genes that represent some kind of a true variation between these conditions. So here you have condition 1 and condition 2 between different samples, and you only want to select genes that represent some kind of a difference. How do we find those genes? We find those genes by looking at log fold change. So fold change represents by how much are those conditions different, and the adjusted p-value. The adjusted p-value allows us to differentiate between statistically noisy and clear data. So we know where's the signal and where's the noise. What is the noise? Noise is the internal variation within that group. So if within a group there's a lot of high and lows, then maybe this is not truly a condition. Maybe this is some random collection of samples. They don't really represent a true change in that condition. And so those are some of the topics that are described in Transcriptomics 2. As a result, we can also visualize specific genes that are responsible for those different changes between conditions. And they also utilize elements of clustering to help us understand how those different genes are organized within some kind of a comparison. Beyond that, when we talk about different patterns, right? So as you saw here, the pattern could have a lot of variation within it. We can make some general assumptions by forcing this to be only representative of 2 conditions, for example. But in so many cases, once we get beyond small numbers of samples, we start seeing a lot of variation. And that variation is going to be based on these more subtle variations within gene and within sample expression levels. And then we can start applying machine learning to study those different patterns. And some of the things that we can use machine learning is for exploratory data analysis, where we do principal component analysis and visually see how variance in data helps describe different samples. We can use it for clustering, where we want to study how closely related some samples are, and you can see that it is a lot like the multiple sequence alignment and phylogenetic tree that we did. It also allows us to assign some kind of a distance metric to similarity. We can use it to also develop a model that helps us differentiate between different types of samples or classify different groups. And based on that classification, to select for features or genes, in our case, that help characterize the differences between conditions that we are studying, right? So essentially, this whole process could be combined also in a sort of a pipeline, where we first explore or mine data, we look for different patterns in the data, we then determine those patterns to be associated with some phenotype or a group, right? And then we can use additional methods for classification and feature selection to determine how to best characterize those groups and potentially apply the same model to a completely new data set that might be coming from a completely different source, but nevertheless can contain some similar variation that we've discovered in our data set. So in detail, all of these methods are explained in this collection of transcriptomics courses, Transcriptomics 1, 2, and 3. And I want to pause here real quick to make sure that overall the process of RNA-seq is clear to everyone. You had a chance to review some of these methods online in those asynchronous courses. I did see a lot of your submission of the assignments and also questions, and I think we've been able to resolve most of them. But briefly, if anyone has any questions about this process of RNA-seq, I'm happy to answer some of those questions right now. Okay, are there any other questions? Okay, Babis, that looks in Greek. So it is a little focused, okay. Okay, I'm not sure. Okay, which file output should we use for differential expression analysis? Okay, so we should use the gene expression table, so table of expression. Once we have that table of expression, we can load the table of expression into the differential gene expression method, like DE-seq, and then assign different columns or different samples to a condition, to a group. Okay, but does the software find alternative splicing automatically? Yes. So the process of alternative splicing, Babis, and Claudia, if you could let me know if that answered your question. Babis, for alternative splicing, okay, yes, Claudia, it's just read count, not FPKM or TPM. And Babis, about the alternative splicing. Alternative splicing is essentially just combination of different exons together. Now, when you look at expression levels, you might have one exon that is expressed at 3, another exon that is expressed at 5, and another exon that is expressed at 1. So the alternative splicing method to determine which isoforms are expressed is to find a pattern that it would be consistent enough with what we see in the data to say that maybe one gene is producing 2 different transcripts, and those are expressed at different levels. So, and that is done automatically. That is done within that cuff links, cuff merge, or some of the other tools that are on there. Okay, let me know, Babis, if that answers your question. Okay, Nachir van Gafar. I have my set of results submitted in NCBI. Which kind of files we can take from there? Okay, so we'll look today at different types of submitted data. So we looked already how we can find full genomes, right, on the genome. Those would be FASTA files. We looked at SRA. Those would be FASTQ files typically. And then we also will look at GEO, Gene Expression Omnibus today. That gives us a table of expression. Okay, that will be a table of expression. Sometimes we need to work with that table of expression a little bit before we use it for analysis. Okay, Wilfried, will these platforms be still accessible after the end of the course? Yes, so for the next, I will have to look exactly again when the program ends, but for the next couple of weeks at least, the platform is there for you to continue working on your independent project. The courses are going to be available as well if you would like to complete them. And after the end of the program, we have a number of different options for you to continue using either the educational or the research platform, and we're happy to discuss with you what the options are. Topi, I do not always see all expected result outputs during my Transcriptomics 2 and 3 exercises. So Topi, it would be great if you can point out some specific example of what you were not able to find. Some of those have changed over time, and I think majority of them are still there, maybe hidden away in some additional folders. So I'd be happy to look into that and clarify and also make the changes to the courses in case that is needed. Okay, all right, Bobby, so yes, I'm happy that I recognize that. All right, excellent. So everybody got their questions answered so far for that. So how do we apply gene expression to the study of viruses and viral proteins? You saw in previous sessions how just taking one protein and analyzing that protein structure can help us understand potentially what different domains that protein has, and we call them functional domains, right? So those functional domains, for example, specifically that we focused on, were either a site that separates the protein into two different parts, or we even talked about very specific functional domains like the receptor binding domain. And we saw how these different elements in the single protein can have multiple different functions after transformation. Overall, how do we know all of these things? Well, the protein function is an area of research that could be achieved through multiple different types of methods. Essentially, we can look at amino acid sequences where we compare sequences, and maybe there's a protein that we know and another protein that we don't know, and we can find how it is similar to different proteins. We can also look at 3D structures of proteins and infer from the 3D structure what might it do or what proteins might it interact with. And another approach is to look at this high throughput data. For example, gene expression would be called high throughput data, or if we analyze molecule efficacy on different cell lines, that would be another example of high throughput data. So when we look at amino acid sequences, we really can look at sequence similarity, genomic context, or feature-based, right? For example, we can look at motifs. Those motifs could have some significance. We can look at global or local sequence similarity between different amino acid sequences. And we can also look at genomic context, right? So where are they placed? When we look at 3D structure, we can analyze 3D structure similarity by looking at how the 3D structure can be aligned to different other proteins inversely or exactly the way that we have it, right? It might give us some idea about interaction with other proteins and other 3D structures. And when we talk about high throughput data, we can look at protein-protein interaction. If we capture protein complexes together, we know that they interact. Or if we look at gene expression, we look at different conditions and how those different conditions are comparable, or how which elements compare those different proteins, and then also do network analysis. And so those are some of the things that we'll talk about today. For example, viruses are very efficient at storing information, which means that they have very specific sequences that they can maintain and effectively package within a virion, but individual portions of that genomic sequence could translate to a number of different proteins. So you can see here an example in the original SARS-1 genome, how the different non-coding or non-structural proteins are produced from very specific positions on the genome. And you can see a lot of different proteins here that are encoded overall. It is, you know, these are the known proteins, at least that we do know about. There are NSP1, NSP3, 7, 8, 9, 10, 15, the spike protein, N protein, ORF7A, and ORF9B. And then we also have the PL-Pro, or the NSP3, M-Pro, cleavage sites, and the structures of NSP5, 7, 8, 10, 15, etc. So a lot of these proteins have different interactions between them. So for example, here you can see how proteins connect together. So NSP7, 8, 1, and 8, 2, T1 and T2 form a specific structure, right? And so these structures are used by the virus to interact with a number of different proteins within the cell, including interference with a number of different processes that the cell would employ to attack the virus because it recognizes it, to support the replication, and also utilize and trick the cell machinery into doing whatever the virus needs instead of what the cell needs. So here are some examples. As you remember, we kind of did an overview of how the virus enters and unpackages its RNA, then produces the viral polymerase protein, and then it starts producing these subgenomic RNAs that encode then into different types of proteins. And then it essentially, the objective is to package another set of RNA into this viral protein. So here you can already start seeing how these different types of activities are done in different areas of the cell, right? And one important aspect of this is this nuclear transport cycle. And the nuclear transport cycle is important because this cycle is where the cell can respond, right? So within the nucleus, you have the DNA, and the DNA is constantly used to produce, like a reference, right? It's constantly used to produce different proteins that are important for the cell, right? So this might include different proteins for signaling, different proteins for enzymatic activity, metabolic activity, etc. So all of that reference code for whatever happens in the cell is within the nucleus. So when the virus enters, it is tricking the cell to do something else, leveraging the cell functional proteins as well, and also trying to evade this immune response. And so trying to evade this immune response is essentially blocking some of this material that is coming out from nucleus. So you can see how some of these proteins that the virus produces, when we have portions of them in different configurations, they can start interfering with some of those processes that happen, and that includes affecting some of the messaging or signaling pathways. That includes cytokines like interferon that are affected, and that also includes some of the functional ways that the cell can respond to the production of virions inside itself. So here are some examples. Here's an example from a publication that says release of severe acute respiratory syndrome coronavirus nuclear import block enhances host transcription in human lung cells. This is a study related to the original SARS. And what they did here is they looked at how ORF6, which is a non-structural protein, when that protein is produced, it antagonizes interferon signaling by blocking specifically that nuclear import, so karyofurin-mediated nuclear import processes. Essentially, what they did here is they understood how ORF6 protein can help understand. So if we look at expression of cells at different time points, we can start seeing how the ORF6 protein interacts with very specific gene promoters and impacts the different host transcriptional factors. So you can see here VDR, CREB1, SM8D4, P53, probably recognize that one, and others. And those are critical for the robust antiviral response that the cell should have. So this type of information, so this is a publication, has an associated data set. So let's take a look at this data set real quick. So I'm going to post this for all of you. Sorry, this privately. Let's go to everyone. Okay, so let's go and take a look at this together. Okay, so here you can see that, again, it's important to remember what is this generated, how is this generated, and this was expression profiling by array. What did they do here? SARS-CoV Urbani or SARS Delta ORF6 infections of CALU3 cells, time course, right? So essentially they took a wild type, they took a Delta ORF6, in other words, the whole genome of this virus without the ORF6, and they wanted to compare transcriptomics of 2b4 cells infected with these different mutants. So if you scroll down here, you'll see that they have a lot of samples, right? And they tell you that there's a mock infection, it's by hour, right? So you have 0, 3, 7, 12, 30, 36, 48, 54, 60, and 72. So a lot of hours. And so they did 3 groups, basically: mock, wild type, and Delta ORF6. So by looking at time course, how this infection progresses, we can start looking at gene expression of those cells and compare those different gene expression patterns to understand differences. Okay, so so does it kind of make sense what exactly they did here? Did anyone have any questions about what they did or how the data was generated? Okay, so thinking about RNA-seq, what is a way for us to summarize what happens here, right? What is some method that you have seen or potentially know from your past experience that can help us summarize what is going on between these different samples overall? Okay, DEG, or differentially expressed genes, suggested by Athanasios. So differentially expressed genes is a good idea, right? We can look for differentially expressed genes, but before we do that, because differentially expressed genes would require for us to select some group of samples from here, right? And we have a lot of different samples. We have here, how many in total? 99 samples. So before we make it specific, okay, so Nachirvan is saying PCA, okay? Claudia is saying build the transcriptome mapping to genome, etc. Yes, we need to build a transcriptome, but once we do that, we'll have thousands of genes for each one of these 99 samples. And the question is what to do then, right? So Nachirvan was suggested principal component analysis, and I think that is a good direction for us to go. So what does principal component analysis actually do? Principal component analysis will use all of these different samples and all of the genes that are included in each sample and show us variability between these different samples. Okay, so let's take a look. This is principal component analysis. The green dots are delta ORF6. The yellow ones are mock infected, and the wild type are the wild type, right? The full genome. And so here, what you can see is that at 0 hours, right? So before infection started, you can see here that all of them grouped together. This is, by the way, only the 0, so only 3 groups after infection. So before infection, they all look the same, right? That's what the principal component analysis shows us, is that before there's not really a lot of variability between them, they're all mixed together. But as they start getting infected, right, they start spreading apart. And so at the end, we can see that a lot of these are separate, right? They're in separate positions on this principal component 2, which is the y-axis, which is not the most significant, right? So the most significant here is principal component 1, which is the difference between non-infected and infected, which makes sense, right? But after infection, there's still a large portion of genes, right? And we see about 10% variability that could be explained by the differences between that ORF6 being present and ORF6 not being present, right? So then we can start thinking about how to prepare that differential gene expression comparison, where we have 3 groups essentially, right? But we already know that the mock is different from both of them. And we can forget about the 0 hour samples because they don't really have a lot of variation there. And we should start looking post-infection, at some point post-infection, right? Select those samples and then look at differentially expressed genes. And here you can see differential gene expression analysis between DORF6 and wild type. And you can see the different hours, right? So this is 60 hours, 48 hours, and 72 hours. So the later hours compared between those two. And now we can find groups of genes that are that could be combined in a specific pathway, right? And so what we are interested to find is how different genes are expressed over time and also which pathways are modulated. So how are they differently expressed between these two different genomes? Does this type of analysis make sense, or the logic behind this kind of an analysis make sense, or does anyone have any questions about this? Okay, as you're thinking about your questions, I'm just going to go back to here and show you where is the actual data and what could be done with the data to analyze it. So here you can see the expression values right here. You can see it's the biggest file. It's or actually not the biggest one, but it's got 16 megabytes. And you can click here on HTTP. Again, I'll just show you also some of the challenges because this is not Illumina RNA-seq, but this is actually a panel. But let's take a look at this data set. So this data set is produced by an array, which means that specific sequences are in each cell of the array, and whatever binds to that cell is counted. But and so the biggest problem is these things right here, right? So you don't see actual gene names. You have to know the translation between this code of the panel with the actual gene symbol. And so that will require a little bit of time. If you see here all the way, there's about 33,632 genes, so a lot of genes for each sample. And here you have the sample names, right? So here you have the sample names, and you can see that they are separated into delta ORF6, and then by hour, and you have multiple replicates for each hour, 3 replicates per hour. And if you scroll down, there's mock, and then there's wild type. Okay, so again, we start with a table of expression. We can then use this table of expression to run principal component analysis, understand how the samples overall differ, select specific samples from here, and run differential gene expression. And then one of the key challenges is a challenge not related to analysis, but related to the technical mechanism that this data was produced. So how do we actually identify what genes are these, right? Okay, so that's a brief overview of an example, how we can use, right? So what does this method allow us to do? It allows us to study the protein function using high throughput data. Okay, yeah, so you cannot actually use David to do this. You have to find the specific panel that they use, the specific array. So you have to find profiling by array. And somewhere here, okay, so this is the platform that they used. And each one of these has a specific naming to convert those to gene names. Okay, these are going to be probe names. So you have to use an annotation process to convert those names. Okay, so that's just one technical difficulty, but the idea here, right? So the idea of finding a data set that can help us understand protein function. We have gene expression data using high throughput sequencing. We can then compare those conditions and identify what is the protein function. When it is present, it has a certain function. When it's not present, that function is not there. And if we look at the publication, in the publication, this is what they found, right? So they found our data demonstrated that ORF6 protein expression attenuates the activity of numerous karyofurin-dependent host transcription factors that are critical for establishing antiviral responses and regulating key host responses during virus infection. And they also confirmed their results by proteomic and chromatin immunoprecipitation assay analyses. What is proteomic and chromatin immunoprecipitation? Chromatin immunoprecipitation means that you have antibodies that connect to specific types of proteins. So if once they found the specific genes that are modulated, for example, present or not present, you can design an antibody that will connect to those proteins or protein complexes even, and then take only those proteins out and then do mass spectrometry, for example, or look at specific binding of those antibodies to a position on the genome and analyze which proteins those were. And by that way, you can confirm through proteomics or immunoprecipitation which specific proteins are actually produced without dealing with, differential gene expression overall, where you do have a lot of noise. So that's a way to confirm those results. The data strongly support the hypothesis that viral antagonists of nuclear import actively manipulate host responses in specific hierarchical patterns, contributing to the viral pathogenic potential in vivo. Importantly, these studies and modeling approaches are not only provide templates for evaluating virus antagonism of nuclear import processes, but also can reveal candidate cellular genes and pathways that might significantly influence disease outcomes following severe acute respiratory syndrome, coronavirus infection in vivo. Okay, so I think a very interesting study. We have that data, we can find that data, we can analyze that data. I hope that this gives you some ideas on how you can think about a project of your own. What are some similar things that could be found based on same kind of an idea? Here you have another publication, Severe Acute Respiratory Syndrome Coronavirus Protein 6 mediates ubiquitin-dependent proteasomal degradation of NMYC and STAT interactor. So there's the STAT pathway, the interferon signaling pathway. You can see that those viral proteins, again, that we don't know what their function is, have been studied again using this kind of an analysis to understand how it interferes with specific interferon signaling pathway, right? So if the cell wants to enable some kind of an immune response, the virus can use these non-structural proteins to interact with that. And here you can see the other side of it, where you have an ability to discover how P53, which is a host protein, how it regulates SARS coronavirus replication and is targeted by the SARS unique domain, and PL-PRO via E3 ubiquitin ligase RCHY1, right? So highly pathogenic severe acute respiratory syndrome coronavirus has developed strategies to inhibit host immune recognition. We identify cellular E3 ubiquitin ligase ring finger and CHY zinc finger domain containing 1 as an interacting partner of the viral SARS unique domain and papain-like protease, and as a consequence, the involvement of cellular P53 as antagonist of coronavirus replication. Okay, and even further, they did a more detailed analysis here. Okay, so a good question, where to get the data for the paper? Okay, so for example, if you have a paper, let's see, this one, NCBI, that's why it's a good place to look for things, will actually help you find some of these data sets. Okay, so you can see this paper. And within this paper, it depends, again, what exactly did they do here, right? So you do have to look into, what exactly they did. Not all the papers are going to utilize next generation sequencing necessarily, right? These, for example, what they did here is they did some analysis of cell culture and expression by GFP, right? So a different way to interpret what is being expressed. But if you do want to find a data set on something like this, I'll give you some examples here in a second. Oftentimes, once you determine what your interest is, right, and that's what we want to focus on today, is how do you go from having an idea, right, having some biological understanding of the different processes involved, and look for data that can help you make that analysis your own. So let me walk through some of these slides, and then we'll talk about, actually finding some data that you can base your analysis on. Okay, so we talked about a couple of these other examples. Okay, let me pause here. So Bobby's is saying, if we found a good set of transcriptomics data, so gene expression of cell lines we want to use, but they're not raw data, we can only find process data. Is there any way to incorporate them in our analysis, or we just need to find a different set of data? What would you recommend to do? So yes, sometimes you will not have raw data available, only gene expression tables. If those gene expression tables are in a specific format, most likely you can use them, and that is fine. The reason why we say it's good to find the raw data is because on one side, when you present your analysis result, it's good to show how you did the analysis from the very beginning all the way until the very end, and that is more for you. If you want to just understand a biological process, then you probably want to just make sure that the data that you take, right? So for example, in this previous example, when we have this array data, I don't know what this data is in, right? So this data is from an array, but I will have to dig in to find out exactly what is this quantification method that is being used to produce these numbers. And that might have some impact on the types of analyses that I would like to do. And that's the reason why it's easier sometimes to find the raw data. Okay, from Grace. Can we plan the project with preprints? Yes, definitely. Preprints give you a framework on how to think about the data set, but also provide you with all the metadata. How did they generate the data? What is the experiment design that they had? Who processed the data, etc. So that's the information that you need to guide your analysis. And so you can definitely start from a preprint. Okay, so we talked about different proteins, right? How to characterize those proteins using high throughput sequencing, what types of ideas we can get out of them to understand how the different proteins of the pathogen interact with the proteins of the cell. Typically, we do look at specific viruses or similar conditions, but sometimes, here's a good example, we can study different features associated with disease. For example, we know in coronaviruses that ACE2 expression is important, and cytokine pathways are also important. Sometimes those could be studied from similar conditions, right? So here you have an example. We present evidence that viral respiratory infections are potential mechanisms of ACE2 overexpression in patients with asthma. Okay, so this is a study that is not focused on how, right? So up until now, we were asking the question, how does the virus cause infection? Or how does the virus, for example, interact with the host proteins? Here's a different question. The question is, could there be a factor with patients, potentially even caused by other viruses? Okay, here we're talking about rotaviruses that are that don't cause severe condition, but this is more a study of interaction between various viruses, right? What if overexpression of ACE2 and susceptibility to COVID-19 infection is actually a result of other respiratory infections, and other respiratory infections that can cause ACE2 overexpression and modulate in some maybe minor way the cytokine pathways, the interferon pathways? Could they actually cause risk, higher levels of risk? So here you can see that they pair design experiments across discovery and validation cohorts of in asthmatic patients The biological replicates treated with rotavirus strain A and C were compared to non-treated. And so here again, you have PCA of this data, you have control group, and then RVA and RVC. And so what might you be looking after you see this separation, right? What might you be looking in the actual gene expression data to find? Who can kind of suggest, you know, what next? What pathways would we be looking for? What genes would we be looking for? Okay, let's take a look at this together. Again, this is available here, you can see, as SRA. So we can download, okay? Nechirvan is suggesting housekeeping gene. I'm not sure that housekeeping gene is what I would be looking for, but here you can see this is by Illumina, right? And you have the different samples, so you can actually start from the very beginning, or you can start with the actual process data already, and you can see that this is RSEM, discovery validation, okay, produced by RSEM. So this data set is probably going to be useful for us to, you know, ask, yes, housekeeping gene. What is a housekeeping gene? Housekeeping gene is a gene that we know what its expression should be. So if they report here ACE2 overexpression, we should probably look for ACE2, right? If they report cytokine surge and, you know, tissue damage, so we can look for pathways related to cytokines. Again, the JAK2, you know, STAT1, interferon pathways that are probably going to be impacted, right, between the different viruses being present, RVA and RVC. Okay, here's another example. This is a publication in Cell. Imbalanced host response to SARS-CoV-2 drives development of COVID-19. What's interesting about this data, let's take a look at this data together. So here's a data set, a rich data set that you can find a lot of different types of data to analyze and play around with. 110 different SRA experiments. Again, if you read here, what did they do? Here we offer an in-depth analysis of the host response to SARS-CoV-2 as it compares to other respiratory infections, right? So we saw SARS-1 and the host response. Then we saw some risk factors maybe associated with other viruses infecting specific types of patients. But what if we start comparing different viruses and see how they cause different types of immune responses? For example, what they did here is treat different types of lung cells. So you can see lung epithelium. You can see different treatments. Lung alveolar, and even they made these cells express human ACE2, and even tested here a specific drug. So if you actually look at SRA experiments, let's look at the SRA experiments and I'll show you some interesting, and by the way, this data set, we prepared some description table. Let's take a look at that. Okay, well, this is just for the SRA links, right? We will have a full data set that you can use, and you can actually find it right here, right? So if we send this to file and do run info, create file, we'll get all of the samples. Okay, and here you can see exactly what types of data sets are available in terms of the SRA, right? So there's a lot of links here. There should be much more. Okay, yeah, so 330, 329 SRA files. Obviously, you would like to select some files before you work with all of these. So you can go to send results to run selector to see some of the metadata about what kinds of what kinds of samples do we actually have here. Okay, so let's change this to CSV. Okay, so here you have more information. You have here treatment, tissue and cell type, and also time after treatment. So here you can see mock treatment, IAV. So these are different types of viruses treated with interferon beta, SARS-CoV infected, mock treatment, RSV infected. Okay, so a lot of different types of viruses and conditions being affected here. And also you can see what types of tissue is available, as well as here, these are not just human, but also ferrets. Again, you can see here by their species. So ferrets, different ages of ferrets, different types of cells, okay, expressing ACE2, etc. Okay, let me answer some of these questions. Could they be looking for gene expression of apoptotic pathway in the previous study? Well, yes, P53 is involved in apoptosis. So, you know, apoptosis and these different genes involved in those pathways could explain what actually happens, right? And by the way, yes, this is what I was looking for. Ruxolitinib is a drug that is being tested for COVID-19, right? So here, I think the point is that a rich data set that you can start selecting specific data sets from to really build a comprehensive analysis of how the host cells might be responding to COVID-19 infection and how that infection might be different from other viruses, as well as trying to understand how potential treatments might have an impact and how different even host factors can play a role, like ACE2 expression that is being induced. Claudia is asking, how are we going to create a single file of the samples we are interested in within a bioproject to use as input to run pipelines? Excellent question. Okay, so here you can see that we have, a huge variety of different data sets of different files, right? And you have specific codes for each one, right? So this is like the ID of that cell, or of that sample. In this file, where you have the links to the SRA, you have the same ID, right? So from here, you select the ones that you're interested in. For example, just the ones that were treated versus non-treated. And here, you select those and create an SRA run info to upload to the pipeline. And I'll share a video on actually how to practically do that. Okay, raw data coming from microarrays and next generation sequencing can be generally treated in the same way in our analysis. Yes, that is correct. Overall, they both show expression of genes. RNA-seq can give you more idea about isoforms and novel isoforms. It also has a better, more reliable level of expression because the microarray can be biased based on what exactly is the sequence used in that microarray and how many variations of that same sequence could be there. And also the method of detection is not as reliable as for RNA-seq. All right, so these are some examples. You can read more about this publication, and we also include that in the Dropbox folder for publications for this session. So you'll be able to find the PDF in there as well. Finally, we do have an example that is non-COVID related here. You can see, for example, similar strategy employed by Ebola virus, evasion of interferon response by Ebola and Marburg viruses. So both of those are very, they cause death, but Ebola is much more pandemic, it spreads much more. But here you can read a publication on how a specific protein, so proteins VP35 and VP24, block host interferon alpha and beta production and inhibit signaling downstream of the interferon alpha and beta and the alpha gamma receptors, respectively, and also VP35, which is a component of the viral nucleocapsid complex and plays an essential role in viral RNA synthesis, acts as a pseudo substrate for the cellular kinases IKK epsilon and TBK1, which phosphorylate and activate interferon regulatory factor 3 and interferon regulatory factor 7. So you can see how a variety of different viral proteins that the virus can use as structural proteins and in non-structural proteins can interact with the different pathways. And so you can see how the differences here between Ebola virus and Marburg virus. You can see here the Marburg virus VP24 protein does not detectably block STAT1 nuclear import. And unlike Ebola virus, Marburg virus infection inhibits STAT1 and STAT2 phosphorylation, right? So they can have different ways to interact with the human response to viral infection. And those types of differences are going to be important. This is, you know, the same family of viruses. So we expect them to function a similar way, but nevertheless, they do not. And yes, here you have even a list for those of you that are interested with Ebola viruses that we did discuss in the context of the program. A good collection of data sets that you can find on the host response to Ebola virus. So let's just, oh, sorry, pasted that to someone private. So here's a link. Therapeutics of Ebola hemorrhagic fever, whole genome transcriptional analysis of successful disease mitigation. So this is for treatment. Conserved transcriptional response to intranasal Ebola virus exposure in non-human primates before onset of fever. Ebola virus species associated with differential pathogenicity induced distinct host responses in human macrophages. Transcriptional profiling of the immune response to Ebola infection. And comparison of transcriptomic platform for analysis of whole blood from Ebola-infected cynomolgus macaques. So I think it addresses a lot of the different questions that we discussed here. Okay, so to summarize, high throughput profiling of protein function and host response is making a lot of advances for high throughput screening of different compounds, repurposing of different drugs, and really understanding how these proteins and how these viruses cause infection. Here are two reviews. One is a description of network-based drug repurposing, how you can take specific drugs that have, drugs target specific proteins. Those proteins should be important to the way that the virus causes different responses at the host level. And so what you would want to do is connect the host transcriptome that is related to infection and the drug-induced transcriptome, right? And there is some overlap between them where you can start understanding if a drug the whole idea of drug repurposing is from, you know, the idea that you already have drugs on the market that have certain effects on the host transcriptome, right? And if the same transcriptome is being affected by the virus, could we try and repurpose this existing drug and see whether it can fix some of those changes and make the infection not as severe, helping the immune system potentially overcome this infection. So some of this, some of these ideas are also described in this review article, How to Discover Antiviral Drugs. So choose viral or human protein targets, molecular dynamic simulations, virtual high throughput screening, docking of different compounds, ranking them, and then also this part of it, right, is related to this right here, experimental testing of top ranked compounds and really understanding at the molecular level, how do those drugs actually work and whether they affect the same pathways. And the pathways contain multiple targets. Not all of these targets are direct targets by specific antiviral chemicals or compounds. They could be affecting them indirectly, just like we saw what the virus does with its proteins. It actually affects indirectly some of those signaling pathways, causing specific evasion of the immune response. So great articles also included in the folder for today, and hopefully would be useful for you to think also about some clinical applications of some of these mechanistic studies that we've described up until now. Mechanistic in the sense that they describe some mechanism and not always necessarily have a direct clinical application. Okay, so are there any questions about some of these examples and examples of how protein function and host response could be studied? The servers allow us to create those expression graphs. Well, yes, so you have network analysis, different shades of colors. Yeah, so you're probably, are you talking about these kinds of graphs? Yes, yes, so you can do this. Okay, so let me address some of those questions. Right here, you have utilities, and you have heat maps right here. So that's called a heat map. You need to have a table of expression. Don't use all of your 33,000 genes. You're not going to see anything, right? So instead, what you can see here is, I don't know, 50 genes, maybe the 50 top differentially expressed genes and across samples. And also this heat map button is going to generate an R script. If you want to modify how it looks, it's very easy to do inside the heat map script. Okay. How can we use Geo for pipelines? So just like I showed, Geo for pipelines, you download the Geo. So you download that data, right? So just like we saw down here, this was just downloaded directly from Geo. So this is already a table of expression. Once you convert this to actual gene names, or if the Geo file was generated by an Illumina experiment, you will already have ensemble IDs or potentially even gene common names. So that's how you can use that directly. And instead of going through the RNA-seq section, right? So instead of going through here, you have all the methods for data mining, differential expression and factor analysis, and also have in here different things like normalization, principal component analysis, and all kinds of other things to visualize your data sets. Okay, any other questions? Yes, Geo is already a gene expression table. So it's not raw reads. This is a table that you can do downstream analysis with. Okay, so let me quickly do a review of independent projects. Nucleotide mutation correlation could help us identify pathways between proteins. Well, so some mutations are going to result in change of expression because they're going to be stop codons, for example. Other mutations will cause a different maybe protein to be produced. So it depends on what you mean exactly, nucleotide mutation correlation. Uh, if you have mutation data and gene expression data, you can find correlation between mutations and gene expression, and that will potentially have an impact on different pathways. Now, protein-protein interaction pathways are different than signaling pathways. So there's multiple different pathways here that we can talk about. So I think your question maybe requires more attention. Okay, let's get to, yes, we do have to finish here in just a couple of minutes. And let's try to quickly summarize where we're going with the different projects. Okay, so independent projects. Your target for submitting an independent project, again, this is not very strict, right? We're not in college here. You're doing an online course, so it's really up to you. You don't have to do an independent project if you don't want to. Independent project is really a goal for you. If you have that, you can submit it and get assistance from us to get it farther maybe and answer any technical questions that you might have in the context of this program, right? Once the program is over, what you will not have access to is our tools and our support, but you will still be able to do your own analysis if you wish to. So just quickly, we will give you some data sets and examples we already have, but we will provide you with more guided tutorials on how to utilize some of these methods in the context of different viruses, including the coronavirus, including these different types of infectious diseases like tuberculosis, not a virus, but nevertheless an infectious disease. We have the data sets and explanation of methods for the ultra deep sequencing that we discussed last time. Some defective viral genomes, involvement in the enterovirus D68, a recurring pandemic in kids. So important to find ways that this virus is causing that pandemic. Ebola. Here, again, you have multiple types of ideas or multiple types of analyses that you can do. So before jumping to a specific project, I think having a sense of, the different types of analyses that could be done, that we've done in the context of this program, can potentially give you insights into what you can do and how you can reanalyze the same data or take data from multiple sources, combine it with different methods that are described in different publications that you've seen, and ultimately creating a novel project that is your own, right? So that is the objective of the project. So thinking about that project has to do with some iterative review of available data and of review of publications, right? So once you do that, I think it's important to start planning your analysis to understand potentially how long it might take for you to complete it, right? So when you're thinking of a project and you want to complete the project by the time that the program is over, plan your analysis in such a way that you have enough time to complete it, right? There's a lot of steps here. Each step might take a different time, and thinking about that time is important for you to get there by the end of the program. We do have a number of people that have already submitted projects, and potentially next week, we will announce the different project ideas once we have the time to review them and have those projects listed out on the forum. So if you don't want to work on a project of your own, or you're a little bit overwhelmed with the different possibilities and you would like to work with a team of other participants from this program and together develop a project that is more impactful, I think that's an excellent way to go. So we'll post some projects where you'll be able to say, I want to join that team instead of working independently. That will require for you to start interacting with those other members. So remember how from the beginning of the program, we asked all of you to have a completed profile. Yes, let me remind you how to submit the project. So again, we have the forum. On the forum, there's a link to a form that you have to fill out. That form has multiple fields I've shown before, right? So you can go to the forum. Those fields have to be filled out. It includes the problem statement, the project description. Okay, so if you go right here, project submission and discussion. And by the way, if you want to post that you would like to work with somebody else, you can also post that here. Okay, so here, project submission page, you click right here. And this is, as a member of this program, you should have access to this page. Well, you'll see the form and be able to submit that form to us. And we then review that form and we answer any questions, and we make sure that the project is feasible and has the data that is required to conduct that kind of analysis, etc. And ultimately, you'll be able to start working on that project. So remember how we told you that it's very important to have a completed profile. Again, that profile gives you a sense of what the background of the person is and how advanced are they and what kinds of materials have they already completed and what they know and or don't know yet how to do. We obviously have some other examples of people that have profiles that stand out. And so we expect that most of you will be at around 5,000 points. That's kind of the baseline. Those of you that have done more are already past 10,000 points. We have a number of people like those. But after completing your project, so this is a good example of somebody that has completed a project from beginning to the end. You can see that she has over 43,000 points. So really you gain a lot of experience, particularly during that time when you work on a project of your own. Now, some of you here might be interested in doing more than a student project, right? You might actually want to get involved in research projects. And so I do want to mention that we have an opportunity for you. If you are making significant advances with your project and you're interested and motivated and have the time, we have a research internship that is on a rolling basis. You can actually get into that research internship or a research fellowship, contribute to some projects that are ongoing, and even get involved in projects that will be published at some time or presented at posters at different conferences. Potentially, if you're interested and you want to become a mentor, we have some other programs that are ongoing, and you can get involved in this program to eventually become a mentor there as well. So just a link there from Mohit also on the research internship. If you're interested in continuing to learn about novel coronavirus and the different aspects of processing different types of next generation sequencing data, working with protein data, and doing more in that domain, we have a program that is starting soon, so you're welcome to join that program as well. If you're seeing how this, yes, all of these are online, so you can do them remotely. They require different types of commitments, but they are all online. If you're interested in seeing how this applies not only to viruses, but also to oncology, including oncolytic viruses and precision medicine where individual factors play a role. So we do have a program on precision medicine as well. It's a short, some of these are fairly short programs and not as intensive as this one. Some of them are more involved. So again, if you want to have more information about some of these programs, please reach out to us. This is the last session that we did in the context of this program for infectious diseases. I hope that you enjoyed the program so far. and hopefully you will be able to participate in the project, and we'll continue to interact with you, and we will propose individual times to meet with either you individually or a group of you that are working on the same project to discuss different project ideas and discuss different strategies to help you make it more effective. So I really enjoyed this time interacting with all of you up until now, and we'll continue to look forward to interact with you. And thank you, Serena, and all of you that have joined. Okay, questions. For submission of project, previous data set is necessary. Previous data sets, obviously you need a data set. So I don't know what you mean by the word previous, but you do need a data set to work with, yes. All right, great, and thank you all for the feedback. How to estimate the timeline for this project? Well, a lot of it will depend on how many data sets, how many steps to get that program to a biologically interpretable result. So once you submit your proposal, we actually take a lot of time to review your proposal and provide you with some feedback and talk to you about planning your analysis effectively. So if you can submit it as soon as possible, we'll be starting to review these projects and providing you feedback, and then scheduling a time to interact with either you independently, or if you have a team working with you, then to interact with the whole team and start thinking together how we can execute this in the shortest time frame possible. Approximately when will we have the feedback? You know, we're starting to do it now. I already started looking at some of them. After these sessions are complete, we have a lot more time to start focusing on the project ideas. So, you know, hopefully as soon as Monday, you'll start getting those feedbacks. All right, excellent. Well, thank you very much again for joining, and for everybody else that contributed to this. And we will look forward to interacting with you again soon.